Hi folks! Today we are going to jump into chapter 14, which covers predation, and we'll also get into herbivory, parasitism, and disease. And just as a reminder, we are going to talk about these positive-negative relationships as a symbiosis. So coming back to our table here, our focus for today's lecture falls under predation, this positive-negative interaction between two species, but also keeping in mind that herbivory, parasitism, and disease also fall into this category of positive-negative interactions. So one thing to keep in mind is that one organism in this interaction is going to have reduced fitness, while the other organism is enhanced as it participates in this interaction. So let's define these different categories here. So predation is considered to be the killing and consumption of animal tissue, whereas herbivory is the killing and consumption of plant tissue. So this koala bear here eating the leaf, the bear here eating the fish. And then on the other side here, we have parasitism and then pathogens and disease. So we define parasitism when an organism lives off of a host. So in this example, we are looking at a tarantula and a wasp here. This is called a pompilid wasp. In the family Pompilidae. And this wasp is considered to be a parasitoid. And parasitoids are typically insects that lay eggs on living hosts. So in this example, the pompilid wasp is a parasitoid and interacting in what we call parasitism. So just some interesting ecology about this parasitoid wasp is that it will, this is the female here, and it will lay its eggs on spiders. We have pompilid wasps here actually in northeastern Ohio, obviously not parasitizing tarantulas, so different species, but what they do is that the female injects a neurotoxin through her ovipositor, and then she drags the spider or the tarantula into a burrow. And sometimes they'll even fly several hundred feet with them, which is impressive looking at the similar body size that you're looking at here. And the spider is still alive, but is paralyzed from the neurotoxin that it injects. Once in the burrow, the female will lay her eggs on the host. When the eggs hatch out, they will feed on the live tissue of this paralyzed tarantula. And then we have pathogens and disease. So here we have a variety of pathogens that we can talk about, from bacteria to parasites to animals, and then non-living organisms like viruses or even prions that are disease-causing agents. So in this example that I'm showing you here, you're looking at a bat that is suffering from what we call white nose syndrome. And you might also notice some white on other parts of the body. But white nose syndrome can attack more than just the nose. It's a fungus that is starting to decimate bat populations in areas where it's been introduced. So I want to first break down parasites into two different groups. We have what we consider to be ectoparasites, and these are parasites that feed on the exterior surface of an organism. So the example that I'm showing you here, getting back to our sea lamprey that are attaching to the outside of this lake trout and feeding on the internal fluids and parasitizing as an ectoparasite. Endoparasites live within the bodies of their host. So the example that I'm showing you here, this is a parasitic flatworm. And there are multiple steps in this parasitic relationship in the life cycle. So at one stage of its life form, this parasitic flatworm, Dicrocilium dendritiginum, is living in its juvenile stage within ants. And the ant is considered to be an intermediate host. And what happens within the ant is that it can infect their brains in a way that's not 100% yet understood. It causes some body color changes as well, but the behavioral change that happens is it causes them to migrate towards light, so a form of phototaxis. And they climb to the top of grass blades where they can be picked up by grazing mammals. So as this deer picks up this infected ant, the mammal is our determinate host or definitive host, as it's sometimes called. And this is where it enters the adult stage. So this flatworm will go through different types of juvenile stages, reaching adulthood within the definitive host. When it gets into the definitive host, it infects the bile ducts and liver of these grazing mammals. So this can happen in deers, cows, horses, pretty much any sort of grazing organism within its range. And sometimes we even see another intermediate host of snails. So snails will eat the slime, ants will eat the slime of the snails and become another intermediate host where they're transferring eggs within their slime to the ants. But these are all living within the bodies of their hosts, whether they're definitive hosts or intermediate hosts, so being an endoparasite. So I'm showing you the full life cycle here. You don't have to remember the juvenile stages, the names of them. So you're seeing a lot of terms here like Myristidia, Cercaria, for example. But again, what we're seeing here is that we have our intermediate hosts here of the snail. Eggs are ingested by a snail intermediate host. Their juvenile stage is released from the snail in their slime. This life stage, which we call a cercaria, develops after being eaten by the ant, so the ant eats the slime. And then our hosts are eating the ants. And why I'm pointing this out in a different slide here is because it can also infect humans. And what we see is in humans, it's usually not fatal, but can cause some digestive systems. And this has been picked up by some humans where ants that are infected have gotten into drinks, water bottles, for example, and then swallowed. So with parasites, typically they're not going to kill the host because they have to keep that host alive in order to complete their reproductive cycles or their life stage cycles, which is very different than disease-causing agents, which can kill their host. So the example that I want to bring up today includes a fungus that we call chytrid fungus. And this is what it looks like in its aquatic stage. And this fungus has become particularly problematic in amphibians and appears to disrupt gas exchange and also can damage the nervous system. So here you're looking at some sloughing off of the skin here that happens as it disrupts gas exchange. And then some infection here that you're seeing. This bright red color isn't natural in this type of frog, but being shown as a fungal infection. So it's thought that it originated somewhere in Africa and has been transported as an invasive species to other places across the globe that you're seeing here in some of these other pins, and has become a worldwide epidemic for amphibian species, leading to some massive declines. There's also some interesting ties to climate change, where we're seeing that in areas where we're getting increased cloud cover, especially in the tropics, we see associated decreases in water temperature that facilitates the growth of chytrid fungus. So it does really well in cold environments, but can tolerate tropical habitats, but seems to be more prolific under colder conditions. So as you can imagine, diseases can have really big cascading effects throughout ecosystems and throughout communities. So the study that we're looking at here is representing the effects of chytrid fungus in what we call pre-decline versus post-decline. So before introduction of chytrid fungus, and then after introduction of chytrid fungus. So there are a lot of letters that are represented here, and I kind of want to set you up to understand how these figures are working. And before we get into the nitty-gritty of the results of the study, what I want to point out is that this study focused on tadpoles as a consequence of chytrid fungus. So the outside circle that you're looking at here represents different primary producers, so species of different primary producers, but also different species representing plant detritus. The inside circles represent grazers overall, so tadpoles and insects. So in the most inner circle, we're looking at different tadpole species. And then on the outside of the circle here, we're looking at mostly insect grazers. And they're breaking this up into studying it in different habitat types, so pools versus riffles. And what you're noticing here overall is that we see less linkages in the food web. So each of these lines are showing food web linkages between species. And you may notice it's really hard to see a lot of linkages here, still hard post-decline. But essentially we have less grazers post-decline in both habitat types and less linkages. So in riffles, we see that 60% of the linkages were lost, and 66% of the linkages were lost in pools, the 60% here being in riffles, due to chytrid fungus. So the main point is that we can see some really large effects of disease on just a few species. So chytrid isn't necessarily affecting our insects, but it is affecting the tadpoles. And we can see breakdowns in food webs occurring due to disease. So the bottom line here is that predator-prey systems are dynamic, and they change throughout time. And whether we think about these predator-prey systems being true predators and prey, we can also expand this to think a little bit about how parasites and pathogens may disrupt communities and populations. So here what we're doing is looking at an example coming from a study that was done in Sweden. And it's showing you the spread of mange from 1975 to 1984. And you can see here we have our first case of mange and the range of that being pretty minimal. And then spread throughout Sweden over time. So here's a poor fox suffering from mange. And what we're seeing here are dates from the mid-1960s all the way up to 1990. Number of foxes harvested and number of hares harvested. So often what we're going to see when we have these long population trends in predator-prey dynamics is that some of these graphs are actually captured by hunters that report their catch. So here we're looking at a time frame prior to mange outbreak, and then a period while mange outbreak is happening. And what you're noticing here is that these predator-prey systems between foxes and their prey of hares is being affected by this parasite of mange. So we're noticing high rates of foxes prior to mange outbreak, low levels of hares. You still see some population fluctuation over time, which we're going to talk a little bit more about. But as foxes become infected with mange, we start to see this skyrocket of hares being released from this decline of predators over time. So the point here is that, one, predator-prey cycles are dynamic, but that they can also be heavily influenced by parasites and pathogens. So in our next unit, what we're going to do is get into predator-prey systems. And at this point, I need to pause our video, and this will be part 1 of predator-prey interactions and thinking about predation, herbivory, and parasitism and disease just in general. And the second part of the video will pick back up with this lecture slide and continue on with the rest of the lecture notes.